So hello, everyone. Welcome to all of you, and thank you for being here today. And it is my great pleasure to introduce Dr. Ben Shallow, who is going to be our speaker. So Ben is a lecturer in statistics at the University of Glasgow, and I have the pleasure to know him personally when both of us were postdocs at the same time at the University of Warwick. And Ben did his PhD in statistics at the University of St. Andrews, which was followed by postdoctoral positions at the University of Bristol and at the University of Warwick. And as of 2020, he became a lecturer in the School of Mathematics and Statistics at the University of Glasgow. And Ben's research interests lie mainly in the field of Bayesian statistical inference, particularly MCMC methods, data integration, and model selection applied to problems in environmental and biological science. And today he's going to talk about efficient Bayesian parameter inference for high-dimensional stochastic biological systems using an approximation. So Ben, thank you for joining us, and the floor is yours.
Thank you, Panyata, and thanks very much for the invitation. I'm very happy to be here to present this to you all today. As Panyata said, I'm a lecturer in statistics at the University of Glasgow. This was work that I actually started during my postdoc at Warwick, where Yorgos Minas was also a postdoc at the time, although he's now a lecturer in St. Andrews, and David Rand as well was my supervisor or PI at the time. So what are we interested in? Well, we're interested in modeling biological systems, particularly those types that are common in life processes like regulatory networks, cell signaling pathways that are often found in mammalian cells. And these types of systems are frequently represented by systems of ordinary differential equations, or ODEs, which describe how these different system components react with each other and change over time as a function of perhaps some unknown rate constants or parameters that we might be interested in estimating for some real data. They're complex because they're often highly nonlinear. They're dynamic. The particular ones that we were interested in are actually oscillatory ordinary differential equations. And what we know from the biology is that although these ODEs represent a deterministic component, the biological systems themselves are actually highly stochastic. And initially that was thought maybe because we're just not measuring enough of the important components in these systems, but actually the feeling is that the biology itself is probably a stochastic component to the systems, and therefore we really need to treat these as stochastic systems rather than deterministic, which is what a lot of the work previously was done. Moving on from that, what I was particularly interested in is doing the parameter inference in these systems, but that's incredibly complicated and largely has been restricted to very simple systems with perhaps a few variables and a small number of parameters, because it's often very difficult to do inference in these types of stochastic models due to high numbers of parameters, the nonlinear dynamics of the systems, and the oscillations, which make it quite complex. So motivating example, and this is the main system that we were focusing on, was the NF-kappa B system. So this is a cell signaling pathway system that we found in most mammalian cells, including us humans. So here we have a diagram of a cell. The outer part is the cytoplasm, and then we have the nucleus here in the middle. Each of these ellipses represents a particular molecular structure that we might be interested in monitoring, and that forms a large part of this cell signaling system. The arrows represent reactions that can happen between each of these different model components, and the small letters correspond to some parameters or rates that happen, which we might be interested in estimating these themselves or some function of those from some data. These systems are incredibly important in a whole variety of different biological applications. They've been shown to be incredibly important in inflammation, in cells death and cell regulation. They've also been linked to the body's natural ability to fight cancer. And often they're initiated by some sort of external factor. So this TNF quantity here in this rectangular box is an external signal that comes from other cells. So it might be a tumor signal or a tumor cell that's emitting a particular signal, and that itself is what initiates the dynamical behavior of the system. And these molecules can move in between the cytoplasm and the nucleus. They can form complexes with each other where the molecules join together, and they also produce messenger RNA, mRNA, as well, which is what's assumed to actually inform the body's response to these particular systems. So this model here was produced back in 2018, or actually a slightly earlier version in 2009. This is the system of ODEs that we're interested in working with. There's lots of models being proposed for the system, but arguably this is the kind of gold standard. It consists of 11 different variables. So each of these variables on the left are one of those single ellipses we saw in the diagram. The capital letters correspond to the variables, and the small case letters would correspond to parameters, which represent how much or how frequently these particular molecules react with each other or join together or then split apart. And we might want to estimate some or potentially all of these parameters from some data. Usually the mathematicians or mathematical biologists develop experimental ways of estimating these, or essentially come up with values by sensitivity analysis or some form of checking of the model to see whether it matches the types of dynamics that they observe in the biology. However, statistical inference is arguably what we should be doing to see if we can estimate these from real data. If we run this system to equilibrium and calculate the deterministic solution, this is what we get. So each of these different lines corresponds to the solution of one of those ODEs. We can see that it's very dynamic, very oscillatory. The exact timing and the amplitude of those oscillations vary depending on which particular variable you're looking at. There's a lot of feedback between them, so you'll get one particular variable oscillating, and then that will then impact another oscillation further down through time. So there's a lot of complexity in these models that we might need to take into consideration. And again, this is just the deterministic component, so we haven't even looked at the stochastic part of this yet. The biologists are actually able to collect data on this. So this is some single cell data that our collaborators at the University of Manchester provided us with. And these are multiple cells treated with exactly the same experimental conditions. And what we can see here is that there's quite a lot of stochasticity in the system. So even with the same conditions, generally the first peak that we observe is relatively consistent, but then the exact timing and the amplitude of these subsequent oscillations changes between cells. And that's why we need to treat these as a stochastic system, because indeed that's exactly what we see in the data. And if we look at the model, then we can see that, again, we get these similar types of oscillations that seem to match quite well what we see in the data, which is, at least positive that we're getting some sort of similar dynamics. And the same, some slightly different biological data from the same system, again, matches the oscillations that we see in the data and the model quite well. So we at least know we're roughly on the right track with the particular model structure that we've got, even if we don't know exactly what the underlying rate parameters should be. So that's the kind of background to the systems we're interested in. We're now going to talk more about how we go about stochastic simulation for biological systems. And to this, we look to Markov jump processes. So these are continuous stochastic processes on discrete state spaces. We assume that X is a vector corresponding to the n different variables that we have. So these are the outputs of the individual ODEs in our system. We're assuming that this is a Markovian structure, so the probability distribution over state at time t, conditional on all previous time points. Because of the Markovian property, we assume is only conditional on the previous time point and not dependent on any previous time points that we may have integrated over. Each of our systems, we assume, has capital N different reactions, and each of those reactions, so those could be a reaction between two different molecules to form a third complex of those two molecules, and those are assumed to take place at a particular rate f of j, which is a function of both the state vector x at that time and some unknown parameters theta, which are what we're going to be particularly interested in this talk. And we're assuming that over a fixed time point t, between t and t plus delta t, those rate constants are fixed. We also have some state change vectors, which are these sj's, and these determine for every different reaction j, how does that reaction change the state variable x. So if variable 1 reacts with variable 2 to form variable 3, that reaction will deplete variable 1 and variable 2 by 1. So in the first and second element of Sj, we'll have a -1, and we form a new molecule, variable 3. So in the third element of Sj, we would have a +1 because we're creating a new molecule of S3 in that process. So for every single reaction type that we can have in our system, and this is driven by the knowledge of the biologists and the system of interest, we can determine exactly how the state vector X will change as a function of that particular variable. And from that and statistical physics, we generate the chemical master equation. So this is a differential equation that determines how these probability vectors change through time. And that's going to change as a function of all of our M reactions and how each of those M reaction changes the state probability vectors, which are going to change from X to X plus Sj. And we're multiplying them by the corresponding rate constant for that particular rate. As I said, the Sj's we're going to define for each of our M different rates. And this is an example of a stoichiometry matrix where each of the Sj's correspond to the columns of that matrix and determine exactly how each of those reactions changes the state vector. And in the types of systems we're looking at, they'll generally correspond to +1. Now, it's impossible to use the chemical master equation directly for any sort of simulation, or at least incredibly complex for the types of systems that we're looking at. So we need to rely on approximations to those underlying differential equations to allow us to do any sort of either simulation or, in the converse, in any sort of inference in those. One way we can approximate the chemical master equation is using a Langevin diffusion. So for that, we need to assume that these rate constants are sufficiently close to each other that they're fixed over that time point. So if we treat dt to be small enough, then it doesn't really matter that the state vector is sufficiently close over that time period that the rate constants, although they're a function of x, are sufficiently close to each other that they're not going to be introducing too many errors. And because of that, we can use the Markov-Jont process to assume that these are Poissonly distributed. From that, then, if we assume that the rates are sufficiently large and we have a sufficiently large number of transitions, we can then use a normal distribution approximation to the Poisson, and we can then get the chemical Langevin equation, which we have at the bottom. So this is an SDE, a stochastic differential equation. Essentially, on the left-hand side of this, we have a drift part of the equation. So this determines the general pathway of the stochastic process. And then we have a variance term, which determines how the diffusion or how variable it is about that general trend. Again, we can do some inference or simulation using this, but again, it's going to be quite complicated. So normally we make a further approximation, which we can either get from an first order Euler approximation to the chemical master equation, or using a central limit theorem type approach to the chemical Langevin equation. And we get the linear noise approximation. So what that essentially says is if we're interested in the state variable x at time t, this is now the stochastic version, we can start off with a discrete deterministic version, which is the solution to the set of ODEs, that's xt, plus some variance term, which is going to be scaled by a function of this omega parameter, which is essentially a measure of the volume of the system. So the larger the volume of the system, the less the randomness is going to matter, because everything is well mixed, and therefore we're scaling it by this portion of 1 over the square root of omega. Because of the fact we're making a central limit theorem or a first order Euler approximation, this xi of t is actually a normal or multivariate normal distribution centered on 0, and with some known constant matrices, a C matrix which determines the drift, and a V matrix which determines the diffusion. And because of the normality assumption, these have formal analytical solutions that we can write down and calculate directly. And we can therefore generate a Kalman filter update to do Bayesian updating in inference terms to do the estimation of these states given the parameters. Now, the problem with this is as we progress further and further through time, we're always conditioning on the initial time point t0. So as t gets further and further and further into the future, away from our initial starting points, these normal approximations will get increasingly more and more inaccurate. So a lot of the work is done that we did was trying to calculate ways of making this inference improved over longer time periods of trajectories. So we don't have this problem of the normal distribution approximation becoming increasingly more and more inaccurate. And really the issue is if omega tends to infinity, then we can see that this variance term will tend to 0, and we'll end up in a deterministic case. And when we're in a deterministic case, it's completely accurate for an indefinite amount of time. The problem is where omega isn't tending to infinity, we'll end up reaching this inaccuracy much quicker, and we get these fluctuations in variance away from the standard distributions that we're assuming are taking place. So how do we deal with that? Well, in 2017, Minas and Rand published a paper in PLOS Comp Bio on a method called the phase-corrected linear noise approximation, which attempted to try and deal with these deviations away from the normal approximation as we progress further through time. And what they showed was that if we use this approach, which I'll talk a bit more about in a second, these approximations to the normal distribution remain accurate for much longer periods of time, even though we're always conditioning on the initial time point at time 0. It's also very fast because there's analytical solutions, again, using multivariate normal distribution approximations. And it works very well for large systems like the adjusted Kalman filter update, which means that things are relatively quick compared to perhaps the standard simulation algorithms that are used for these types of problems. So the idea of this algorithm, it stems intuitively from the idea that systems within a attractor equilibrium remain accurate for long periods of time. So what we mean by that is these systems that are very, very regular in terms of their oscillations are often driven by local points that they're attracted to. So the stochastic deviations in those systems can never deviate too far away from this underlying limit distribution. So what they do is they think, well, if we're on this limit distribution, we're constantly going to be accurate, irrespective of how far into the future we're going to predict. Whereas the idea for systems that move away from this limit cycle is that the stochastic trajectories exhibit random walk behavior in the phase space. So these variations and trajectories will increase in order square root of t. So how do we look at this? Well, if we imagine this is the system that we're interested in, and we're looking here at a phase space representation, imagine that we've got this oscillatory system, which is just constantly doing a circular movement round to where it started. This red line in the middle is the underlying limit cycles. That corresponds to the case where omega is tending to infinity. If omega isn't tending to infinity, then you get these stochastic realizations, which we can see in blue. And what the PCLA paper did was they looked at where these stochastic trajectories intersect a transversal S of x to that underlying system. And what they showed was that the location of those points where they interact with this transversal, again, are very well approximated by a multivariate normal distribution. So whereas previously the linear noise approximation is constantly assuming that we're on this red trajectory from time 0 all the way through, and we're just trying to propagate the uncertainty using this underlying system, if we project back onto this distribution here, so where these red crosses are, instead we're using this time point here, we project to the corresponding time on the underlying limit cycle, then this approximation is going to remain accurate for much longer periods of time. So when we do our Kalman filter, we start off at time t0 and we project forward as you would with a standard Kalman filter up to time 1, which is the next time that we have some data. And the standard linear noise approximation would just keep doing that through time. But the problem is that there's these deviations away from that underlying approximation to the system. So what we're going to do is we're going to project back at each of the time points onto the corresponding point on the red line. And that we call S1. And that's the phase, the corresponding phase of that underlying system at that time point. And then we use that as our initial time point for the next simulation forward, and we predict up to time t2. At time t2, we then need to do a correction back to point S2, and we constantly do this correction at each time point to try and correct for these deviations away from the true underlying system. And what they showed was this remained accurate for much, much longer periods of time. So the idea is we iteratively cycle through these standard linear noise approximation steps where we project forward using the standard Kalman filter, but then we're going to correct for instead of using the multivariate normal, approximation as the standard linear noise approximation does, we're going to try and try and correct for that by projecting using this G. So this G projects back onto the underlying correct point on the limit cycle. And because of this multivariate normal assumption, again, we can calculate these conditional probabilities of the state at time ti, given the state at time ti minus 1, and these rate parameters theta that we're interested in. So how are we going to do the Bayesian inference? Well, one of the problems that we had was that these systems have a very large number of parameters. This one has approximately 30 parameters. We didn't expect we would necessarily be able to estimate all of those parameters from data, because often there's a lot of correlation between those parameters. They often appear as products rather than individual quantities, and we know therefore that there's going to be some identifiability issues in the system. So what we wanted to do is to try and work out if we could somehow predict which parameters we might be able to estimate from data before we actually ran the inference and found that it didn't work. So we did that using some system solutions, system analysis using the Fischer information matrix. And the Fischer information matrix measures how much information is contained in X about some parameters that we might be interested in theta. So if we assume that X has a multivariate normal distribution, and we know that we can assume that for the linear noise approximation and the variance that we're using, then the Fischer information has a formal analytical form, which we show here. So we can calculate this in advance of doing any sort of inference. We can calculate the Fischer information matrix for our system, where the mean vector is corresponding to all of the individual outputs of our model. So in the case of the NF kappa B system, that's the 11 variables. And we might have up to, say, 30 or 40 parameters that we might be interested in looking at the sensitivity for that particular variable. So when we did that for the NF kappa B system, we very quickly found we were in a very difficult position, because this model was actually only really sensitive to 2 of those parameters, which happened to be C1a and C1. Doesn't really matter particularly what those parameters are, but for roughly 25 of the parameters we're interested in, the model isn't really sensitive to those parameters. So that's a real problem when we're doing inference. If we try and estimate any of these other parameters, we're likely to have some degree of difficulty, because whatever we do to the parameter, the model isn't showing really any sensitivity to those changes in that parameter. And if we look at an eigen decomposition of that matrix, we see a similar kind of pattern. If this is the log of the eigenvalues of that eigen decomposition of the FIM, and we can see that the first 2 are relatively high, the first 2 ordered sensitivities, and after that it drops off very, very quickly. So this again is in log space, and this really is a very, very steep drop indeed, and then it continues to drop roughly exponentially after that. So what this suggests is really, in the way that we're looking at the system at the moment, there's only really 2 parameters which can be estimated from any data when we're looking at this system. So next we need to think about how we're going to do the formal inference. So from the Kalman filter, we can calculate these successive probabilities and the overall likelihood of a state system at some time point, and the parameters theta that we're interested in is going to be, because of the Markovian property, we can just take the product of the successive likelihoods at each of the time points. So usually these time points would be times at which the data is available, although it's not necessary, but we do usually assume that these TI are the corresponding times that we can interrogate the system because we've got data at that point. So the Kalman filter that we have allows us to derive these, the predicted state at time ti minus 1 given the known state at time ti. We then use the phase correction from the linear noise, or the phase corrected linear noise approximation, to obtain the correct noise distribution, or a better noise distribution than would be obtained under the standard linear noise approximation. And then we use a standard tradition to update the model prediction when we get this new data point T xti. And we successfully do that because of the Kalman filter. These are all multivariate normally distributed, and we just take the product of those distributions over the successive time points to get our overall likelihood. So what we're interested in doing is calculating, as with Bayesians, we want to calculate a posterior distribution, and this is a joint distribution over both the unknown parameter vector theta and the unknown state vector x. X here is just really a nuisance. We're not really necessarily interested in the state trajectory itself. It's really the parameters that we have to estimate, but it's another unknown and we're going to have to integrate over that. So we have a likelihood of the data y given x and theta, and that we assume is some sort of normal distribution with some error that we might estimate. So that's essentially the measurement error. We have the estimate of the state given the parameters. So that's our linear noise approximation Kalman filter. And then we have some priors on our underlying parameters. And again, exactly which of the 27 parameters is going to estimate is going to be a part of the simulation study that we're going to do. Being Bayesian, we want to do this, estimate these parameters. They're highly complex, multivariate posteriors. Although the underlying probabilities are normal, the actual posterior distribution itself isn't, because of the way in which the parameters are linked to the data. So we can't do a formal Gibbs step. We have to do some more complex MCMC sampling to get posterior estimates of these parameters. And we've got to integrate out the states as well as the unknown rate parameters. Now, obviously, a standard way to do this would be to use something like a Metropolis-Hastings algorithm. I'm sure many of you are familiar with, but because of the high degree of correlation within the states and between the states and also the parameters, we sort of expected that Metropolis-Hastings algorithm might not work particularly well straight off the shelf, and we might have to do something a little bit more complex and perhaps look at some more complicated, efficient MCMC algorithms to try and deal with this underlying correlation between the parameters and the states. So the first out-of-the-box method that we used was the Metropolis-Hastings algorithm. The general approach is we construct a Markov chain. The Markov chain we construct has the stationary distribution equal to the posterior distribution that we're trying to estimate. We initialize the chain with some parameter vector theta 0. We update or propose to update that parameter vector using some usually symmetric distribution Q. And then we're able to accept this proposed value theta t plus 1 with this acceptance rate, where it's a ratio of the posterior distributions pi at the proposed value theta t plus 1 relative to theta t, and then we normalize that according to the ratio of the proposal distributions. Now, if the proposal distributions are symmetric, then these two quantities are equal to each other and they cancel out, so we just get the ratio of the posteriors, and that's why we often choose the proposal distributions to be symmetric, but that's not a requirement. And really, it's the choice of Q which is very, very important here, and the efficiency and the ability to estimate these parameters quickly and well is really dependent largely on the choice of Q. So we did do quite a bit of work on tuning and adapting these distributions to try and get the best approximate inference that we could. But I'll talk a bit more about that in a second. Another approach is using a method called parallel tempering. And the idea here is that we run multiple MCMC chains in parallel. And initially, there's no transfer of information at all. So they run independently of each other. The only difference here initially is that we define for each of the C chains, we define a beta parameter between 0 and 1. The first chain always has beta equal to 1, and then subsequent chains have beta less than 1, but greater than 0. We define the energy of the system at the particular iteration to be the minus negative, so the negative log likelihood. And then the acceptance rate, again, very similar to the Metropolis-Hastings algorithm, but this time all we're doing is we're scaling it by this beta parameter. And the idea here is if beta is equal to 1, this acceptance rate will be exactly the same as the standard Metropolis-Hastings algorithm. If beta is less than 1, then you're scaling the posterior by a factor less than 1. So as beta tends to 0, you're tending towards a completely uniform distribution. And the idea is that in the lower temperature spaces, you're much more able to traverse the parameter space more efficiently, although you're not sampling the true posterior, which is the case when beta is equal to 1. You're able to use those samples to try and move around parameter space more freely, and you're less likely to get stuck in local modes of the distribution, which can be very common problem when you've got high dimensional parameter spaces like the ones we're dealing with. And then finally, every so often, so roughly NS iterations, you need to choose this value when you run the algorithm, but you propose to swap the chains around and the values of beta with the corresponding chains. So although the lower value chains are sampling more efficiently, we're not able to use those samples because of the fact you're not sampling the true posterior, but they can be really important in inferring areas of parameter space that you would be useful to explore in the true parameter space. So by switching the chains around and the corresponding values of beta, you allow that information to pass through the chains and potentially move to areas of probability space that you wouldn't be able to visit in maybe a standard metropolis algorithm because of the fact there's some area of low probability space in between that you've got to try and pass through to get there. So these can be really helpful for multimodal distributions, particularly, which is what we'll talk about in a second. Finally, another approach is to use what's called the metropolis adjusted Langevin algorithm. And the idea of this is to try and use the geometric structure of the posterior to move around more efficiently than just jumping around randomly, which is what you generally use with the metropolis. An SDE again, but this time it's centered on the derivative of the log likelihood with some Brownian motion term added on. And if we do a first order approximation to that, then we can get a normal distribution proposal, which uses the information about the parameter derivative or the derivative of the parameter space to try and move around more efficiently. And here, this delta theta, or the upside down delta theta, is the gradient of the log likelihood, and an epsilon is a parameter that has to be tuned to determine exactly how big these steps are along the gradient of the function. And that uses the first order structure. So there's the standard derivative. In 2011, Mark Girolami and Ben Calderhead published a paper which took that further, and they used higher order geometric structures using Fischer information or other Riemann metric manifold metrics. So the idea now is that we're going to consider the posterior space as a very high dimensional function of the underlying parameters. And if we're able to use the information about that geometrical structure in high order space, we can move around the parameter space much more efficiently. And again, we can generate a proposal distribution Q, which is now a function of this high order manifold in high dimensional space. And we can again scale it along regions. So we tend to move through parameter space to areas hopefully of higher probability and avoid regions which are of lower probability. Now, the problem with this is you can see here, we've got to calculate lots of derivatives. The G theta, this Fischer information metric, again, is a function of the derivatives, and we've got to take inverses of those at every single iteration. So whilst it may be much more efficient in terms of moving around parameter space more freely, it does come at a computational cost. So again, one of the things we might be interested in is looking at the relative improvement in terms of parameter inference, but remembering that some of these are going to be much more computationally intensive than others. So in terms of results, we looked at the NF kappa B system. So this was the modulating example that I introduced right at the start. But from the Fischer information matrix that we looked at, we found that only two of those parameters were really important or sensitive, and that was C1a and C1. And that's what we see here. So each of the three columns of this plot correspond to the three different MCMC algorithms. The left-hand column is the standard random walk metropolis. The middle column is this geometric Marlow, which uses the Fischer information matrix to move around parameter space more efficiently. And then finally on the right, we have the parallel tempering algorithm, which runs multiple chains in parallel and then shares information to allow you to move around more efficiently. And what was nice was it seemed that all three of the algorithms converged pretty quickly on the true value, which is this red dotted line in the middle for both the C1a parameter and the C1 parameter. So we were able to estimate the true values. This is from simulated data where we knew the true value, and we could estimate those quite accurately. What was interesting, particularly, was in the parallel tempering algorithm, there seemed to be the second mode of the distribution that it was occasionally visiting very close to zero. And we can see this if we look at the bivariate marginal plots of the posterior. We can see there's a second mode much closer to zero. Although the first mode is roughly centered on the true value, there was this second value that it was exploring. And the other two algorithms weren't really able to explore that very well. There's perhaps a very small one occasion where the Metropolis Hastings algorithm managed to get down there, but it very quickly jumped back up to the main mode and didn't visit it again. So we were really interested in why it was that we were getting the second estimate down towards 0. Was that an error? Was it a problem with the algorithm that was somehow incorrectly studying the posterior or exploring the posterior? Well, what we did was we tried plotting the solution of the system both at the true mode, and we saw that that very closely matched the solution of the underlying original model. And then we looked at the second case where the parameters C1a and C1 were much closer to 0. And what we found was that very closely matched the original system initially. So the first peak very closely matched the same value. But after that, there was no further oscillations in the system. Now, the problem is that we observe this system at discrete time points, roughly every hour, every 60 minutes. And if you look at the time points of every 60 minutes, which happen to correspond to the peaks of the distributions, because that's when the biologists are best able to measure the data. It's most apparent when these are at the height of the peaks. They tend to measure it. And if we matched the peaks of the distribution of this model output with the line here, we got exactly the same values. So what's really happening is the system is doing exactly the same same thing at the time we're interrogating the model at, so the time we have data at, but there's no information in the troughs for it to say, well, actually, this model matches the data in terms of the time points we have data as well as this model did. So essentially what we're saying is that the parallel tempering is actually able to visit the second mode, which is perfectly consistent with the data that we have. So this is an error. It's just the fact that we weren't collecting data necessarily at the time points we needed to. If we also collected additional time points of data in the troughs, then we might better be able to determine between these two different parameter estimates. But in terms of the data we had, this is a perfectly legitimate solution to the model because of the fact it roughly flat lines at the same time as the same amplitude as the peaks in the original model formulation. In terms of the efficiency of the model, so if we look at the random walk MCMC, we can see that it exhibits random walk behavior. It's quite slow to explore the parameter space, and it does get there eventually, but it is quite slow, and it takes ages for it to kind of reach this upper point in the parameter space that we see up here. If we look at the parallel tempering, then it's better able to explore this upper region very quickly. It learns about the parameter space much more quickly, and eventually we'll see that it starts to explore other regions of parameter space that the other models or the other algorithms weren't able to explore. So this is the case where it then drops down to 0, and it's able to do that much more efficiently than the standard random walk Metropolis algorithm was. Finally, if we look at the behavior of the geometric algorithm, now this exhibits more smooth behavior, so it starts to learn about the parameter space quite quickly. And it then sort of, because it's a diffusion-based algorithm, it diffuses quite slowly. So although it's relatively efficient in exploring local parameter space, it takes quite a while to explore the more extreme areas of the parameter space because it's got to learn about that progressively over the course of the parameter learning algorithm. So what we ultimately concluded from that was that although the efficiency of the geometric algorithm was probably better than the standard Metropolis algorithm, really it was only the parallel tempering that was able to explore the parameter space fully. And when we were looking at estimates of uncertainty, then this is going to give much better estimates of uncertainty and credible intervals than models that just aren't able to explore this parameter space more efficiently. So when we moved on to other simulations and other types of systems that we're interested in, we focused almost exclusively on the parallel tempering as the best method to move forward. So the next question was, well, what happens if we add more parameters in? So we know that the two parameters we estimated had high sensitivity, and we were able to estimate those quite well from data. We're now going to rerun the analysis by adding in a third parameter, which according to the sensitivity analysis wasn't actually sensitive. So we wouldn't expect to be able to estimate this very well. When we did this with the random walk algorithm, it actually converged, but it converged on completely the wrong value. So the solid line here is actually the true parameter value that we simulated from. This upper parameter here is the new one that we've added that the model isn't sensitive to. And it actually looks like it's converged, but really it's converged on completely the wrong value. The other two have roughly converged on the true values, but the mixing wasn't nearly as good as when we only had two parameters. We looked at the geometric algorithm again, so this is the manifold Mahler. It seemed to roughly center on, or maybe include the correct value occasionally, but the behavior was very strange and it started doing some very weird things in the middle. Which may suggest that some of the extra uncertainty or lack of sensitivity in the model causes some issues with these geometric methods. Finally, we fitted the parallel tempering. Again, the mixing wasn't perfect, but it does seem to be able to estimate all three parameters quite well. We were slightly concerned about how it was estimating this parameter, third parameter, when, as far as we were concerned, there was no information in the model. But when we looked at the posterior marginal for this parameter relative to the prior, so here the blue line is the posterior marginal and the red line is the prior that we fitted. What we saw was actually what it was giving was just giving the prior distribution back pretty much. There wasn't really any information in the data. It was just that the parallel tempering algorithm was better able to explore that, and having this parameter, which really wasn't sensitive, didn't really cause issues with the model estimation or the inference procedure. But it didn't really give us any benefit relative to what we started out with, whereas the other two algorithms seemed to cause issues when we tried to estimate parameters that weren't sensitive. If we compare this to the parameters that we could estimate, then we can see here there's a huge amount of information gained from the posterior relative to the prior, and this was to say was for the parameters that we had sensitivity to. So again, sort of gave support to the idea of our approach, which is to do sensitivity analysis first to try and get an idea of what we can potentially estimate, and then only really try and estimate the parameters that we can actually have some sensitivity to. If we want to integrate over parameters that aren't sensitive, then the parallel tempering algorithm seems to be the best approach to that, to at least do efficient algorithms where you just end up with the prior distribution back at the end of it. So that was a system where we knew in advance there was only really two parameters we can estimate. We looked at another model. So this was for Drosophila clock. So this is a circadian clock, which is very oscillatory again. It's often usually entrained by external factors such as light or temperature. And it roughly has I think a 20-hour cycle, so roughly a daily cycle in these Drosophila flies. If we do the singular value analysis, so this is again is the Fischer eigendecomposition of the Fischer information matrix, we saw here that there was a lot more large eigenvalues or large sensitivities before it dropped off. Roughly up till 10, there was still some, although it's dropped off slightly, the 10th eigenvalue was still relatively high. So we felt that with this system, we might have a much better chance at estimating more than just two parameters. This is what the system looks like. So again, we've got a large number of variables. It's very dynamic and oscillatory. We've got these feedbacks happening. The timing of the periods or the peaks is relatively slightly more consistent with this system. But again, it varies drastically from variable to variable, how large that oscillation is. But it's a much more regular system as well, because there's less of an external factor be driving this than the stimulus in the previous system. But again, these types of systems have been very difficult to do estimation in previously. So there's 10 variables, 41 parameters. We looked at the sensitivity again in more detail. So we tried to see which of the principal sensitivity coefficients and which of the 40 different parameters that they were most important. So the shading here, very pale and very dark, correspond to large absolute sensitivity, and the mid-level blues correspond roughly to no sensitivity at all. So we tried to pick parameters here. Actually, we picked 12 parameters, which were particularly high in sensitivity because we expected to be able to estimate those more reliably. And when we did, we actually found that we could estimate all 12 of those relatively accurately from a relatively small amount of data. So this was really quite exciting. We've gone from a system where we only estimated 2 because the model itself only allowed us to estimate 2. But if we have a model which is much more variable and much more sensitive, then we can actually estimate as many as 12, or actually possibly more. We didn't push it any further than 12, but this is a really, really large percentage of the 40 odd parameters that we started off with, and is a real positive that we're showing that this type of approach works quite well in systems where you have high sensitivity. So in terms of some further issues, we found that even the adaptive MCMC requires reasonable starting values. The parallel tempering gets away with some of that. If you run multiple chains with different starting values, then often that can allow you to be less sensitive to starting values. And we also need to get good measures of the covariance matrix. When we do the adaptive tuning, we use the covariance matrix of the samples we've generated so far to try and get an idea of the covariance in parameter space. But really, you need to get a relatively good estimate of sigma hat, the covariance in parameters, before you can really use this, because if you don't, then you end up causing issues because sigma will be much too large or much too small and won't give a good representation of the posterior correlation. Yeah, I've said that they're very sensitive to starting values, although the parallel tempering seem to alleviate some of that. In some systems, very small changes in parameters led to very large changes in the system behavior. So particularly with this model, once it converged on the true parameter, it actually didn't want to move very far away at all. So that's why we're getting these relatively poor mixing after that, because even very, very, very small changes in parameters led to very, very different changes in the system behavior. So once it's found that location, it didn't want to move very far away from it at all. But if we try to reduce the proposal variance too small, then it really wasn't allowing to move the potential to move away from that point, which is what we really wanted to do as well. So it's kind of a trade-off between these, as you know, most MCMC algorithms tend to be. We're in a good position with the simulated data that we know what the two parameter values were. So we could start the algorithms off not too far away from the correct values. Obviously, with real data, we might not have an idea of what those parameters might be. So that was something that we kind of looked at more. We tried varying and increasing and decreasing the starting values quite a lot. And for the circadian clock system, it didn't seem to matter too much that it relatively quickly managed to find the true posterior. But in cases like the NF kappa B system, where there's a lot fewer sensitive parameters, this can be really quite a large challenge. Some methods of proposing perhaps running an approximate Bayesian computation first, just to get ideas of which parameters are roughly in the right area, and use those estimates from the ABC algorithm to initialize the MCMC. Obviously, we wouldn't want to do that to influence the prior distributions, but if we're just doing it to influence the starting values of the MCMC chains, then it shouldn't introduce any biases. But again, that needs some further work in how we go about dealing with this sensitivity to initial values. So we're currently pushing this further. We're looking at larger systems again with greater number of significant eigenvalues to see if we can go beyond 12 parameters as estimating for more. We've shown that PCLNA combined with efficient MCMC algorithms allows us to accurately estimate some of those parameters. The geometric proposals were good, but the computational demand was a lot more than perhaps was necessary. The parallel tempering was a nice mix of those two, that it learns about the posterior space quite quickly, but because of the fact you can run them in parallel, you're not really increasing much the computational demand or computational time relative to just a single MCMC change. We're trying to fit this to real data, and there's lots of issues with that in terms of more noisy data. Often the biologists aren't actually able to measure all of the variables in the system. So we're looking at what happens if you perhaps only have measurements on 2 of the 3 variables rather than the 10 or 11 that you actually need, and how that affects the parameter estimation. And we're also interested in the kind of the impact of this further downstream. So often these systems lead to gene expression or hybrid models, and how do we go about using this inference to try and inform those further gene expression data, or see further gene expression models as well. And the idea is to try and move more to an understanding of the information theoretical aspect of signaling pathways to these oscillating systems. Now that we've been able to estimate some of the parameters, we're quite interested in determining what that means and how does the body actually respond to these systems. What do the oscillations actually mean, and how do they inform how the body responds to external factors like tumors and things like that. So just a few references from the papers that I've cited. And I'd also like to acknowledge that the original postdoc work was funded by BBSRC. And I've also received recently an Edinburgh Mathematical Society research grant, which has allowed me to fund some of the computational work that's been done for this talk. So that's it for me, and thanks very much for listening. I'm happy to take any questions that you may have.
So thank you, Ben. That was a very interesting talk. Do you have any questions for Ben? Feel free to put on chat as well or raise your hand. Oh, yeah, Xiao Cheng, do you want to start first?
Ben, thanks for the very, very interesting talk. Can I ask you, you mentioned in the summary that you need to have a good estimate for the matrix and sigma hat. How do you make sure your estimation is good enough?
Yeah, that's a really good question. So, I mean, in terms of the proposal distributions, the proposal distribution is essentially support as the parameters that you're trying to estimate. So really, it doesn't need to be really good. The algorithm will still converge ultimately, even if your estimate isn't great, but it will potentially take much longer to get there. So what we did was we tried some tuning, which perhaps leaves the algorithm to run independently for a while, and once it's run and started to get an idea of which parameters areas of parameter space that it quite likes to explore, we then use that to start approximating the covariance function, and then gradually through time, then the approximation will get better and better and better as the algorithm progresses through the iterations. What we found is if you start that tuning too early in the algorithm process, that the estimate of the covariance is just really poor. So you're trying to force it into areas that it hasn't explored properly yet, or it still won't move out of those regions. So what we did was we tried some sensitivity analysis to try and work out how long you need to wait for. Usually maybe 200, 300 iterations initially for these systems seem to be enough to get a good idea of the covariance function. I'm sure there's more theoretical ways that you could probably look at this and try and as you say, estimate how close an approximation to the true underlying covariance of the posterior is. But again, it's always this trade-off between trying to make something more complex to try and do something more efficient. Is that efficiency going to outweigh the computational demand of these more complex methods? And in some cases, it might well be yes, that it is worth it. But we found that in this case, the parallel tempering was really the best method anyway. So no matter how much time you spent trying to tune the covariance function of the Metropolis Hastings algorithm, it was never going to visit those second modes anyway, because it just couldn't jump across those areas of low probability. So we didn't look too much more into that because we'd sort of already decided that the parallel tempering was the way forward.
Okay, thank you very much.
Any other questions? So I have some. Maybe I can do some, and then feel free to the rest to say. So I was thinking more about the timing. You didn't mention about the tempering algorithm. I think I have a rough idea of how it's working, and I think you need to, like this beta that you mentioned, you need to do it, you need to repeat like quite a lot of times, right? You need to choose how many betas you want.
Generally, the larger the number of betas, we found generally that you get an improved performance, but you're going to be somewhat limited by the number of processors that you have available to you. So again, it's this kind of trade-off between do we maximize, if we can run it on a HPC server with 20 or 30 cores, then yes, it's probably worth having 20, 30 different values of beta. But if you're on your laptop where you only have 3 or 4 available to you, then you're going to be somewhat limited by that. What we did find was that we wanted Obviously, you have to have the value of beta equal to 1 because that's sampling the true posterior. But then if you're limited, then really moving the betas towards the 0 end where you're able to move around parameter space very freely, that was what we found was the most helpful. So if you've only got 2, then don't have one at 1 and 0.9, have one at 1 and 1, maybe 0.01. And, then you've actually got the biggest discrepancy between the two chains and the most gain in the inference potential as well.
Okay. Yeah. So, okay. So 2 probably, how many you use for beta, like in the examples is like.
So I think used 4 or 5.
Okay. Yeah. So it's enough to get.
Yeah,
okay.
so we tried 2 and that didn't really seem very good. You didn't really get much of an improvement. But by the time you've upgraded to 4, it seemed to move around very quickly. And that's, all the results are presented were for 4 different betas. But we could push it further and see if that helps. But I think, for our purposes, that was sufficient.
Okay, thank you. And also, I have a question about if you thought about like semi-Markov model. So far you have like the Markovian assumption, which probably is accurate for your systems, I guess. But did you think about like incorporate this methodology for semi-Markov models in a sense?
Yeah, I mean, there was obviously ways of doing that. It's not something we've really thought about or looked at. I think, a lot of these types of systems that kind of come from the statistical physics literature where the original stochastic simulation algorithm is very based on this fact that, the next time point is really only dependent on where you are now, and well-mixed systems, and that's all you really need. But certainly, and we've kind of thought about other systems, epidemiological systems where you probably get similar sorts of behavior to these. In those sorts of situations, you may well need the semi-Markovian.
Yeah, because I think the methodology is quite general, so I can see it like be applied on epidemiological models, and maybe you need to the semi-Markovian.
Certainly. Yeah, it's not something we've looked into, but it's certainly a good question and something that we could perhaps think about.
Okay, thanks a lot. Any other questions for Ben? Yeah.
Hi. Can you show me? I don't know where is it, but the Langevin equation where you have SF something like that.
Yeah, sure.
Can you tell me what S again?
Yeah, so S is the Stoichiometry Matrix.
Okay.
So the idea of it is it determines how the state vector changes as a function of each of your different reaction types. Okay. So if we go back to our model, we can see these arrows corresponding. So essentially, for example, if we look at this NF kappa B, one of the reactions that NF kappa B can have is it can react and form a complex with I kappa B alpha. And then you end up with this additional new complex, so that I kappa B alpha and NF kappa B join together to form this complex. So that reaction will deplete I kappa B by 1 and NF kappa B by 1 because they've now joined together. And it will increase the I kappa B alpha NF kappa B complex by 1 because you've now formed a molecule with those two structures.
I see.
So for every single reaction type, you need to determine how it's going to change the concentration of each of the variables in your model.
Okay. And what is omega?
So omega is a parameter which represents, it's called the system size. So again, generally,
Yes. Can you stop here? Yeah.
So again, this is it's a measure of how large the system is. So this really comes from the statistical physics literature, where the Van Kamp equations to kind of determine that the larger the system you have, the more well mixed populations are, and the particles in those domains are less random because they're not bumping into each other at random turns. They're only going to be interested in the particles close by to them. So the larger the system is, generally the less stochastic you would expect it to be. Whereas if they're all very tightly compact in a small volume, then they're going to be randomly mixing much more and bumping into each other in different orders. So therefore, the system would be expected to be much more stochastic.
Oh, so very interesting. So it also has very similar meaning at temperature, right?
Yeah, I guess it's a similar kind of idea that the larger the value of omega is, the more the less temperate it is.
And when you take omega to plus infinity, it seems like you take the zero temperature limit, and I don't know, there is a Friedin-Guenzo theorem to study the limiting of it. Is it relevant to this?
I'm not familiar with that theorem, so it could well be, but I'm not sure. Yeah, I mean, it's essentially a parameter that we have to decide at the start of the algorithm, and both the model itself, when we're simulating data from a model, or when we're doing inference as a function of this parameter, and essentially it determines how stochastic those trajectories are. So generally, we find that the accuracy is very, very high when omega is small. As we increase omega, the accuracy is likely to get weaker and weaker. But what we've shown is that with this new algorithm, we're able to estimate things from much larger ranges of parameters than had previously been done with the linear noise approximation as it was previously.
Yeah, I'm quite intrigued by the scaling between the grid term and the noise term. Usually, I don't know, is it a gradient flow because S appears?
Yeah, so it will be. So with this, although this is the chemical master equation, when you do an approximation using what's called the chemical Langevin equation, it does then become a gradient term essentially. So this is a derivative. So this really comes from the solution of the ordinary differential equation, and then this comes from the.
Okay. So SF, like you multiply S times S, or you apply F to S. I don't know. Okay, I see. Okay, yeah. Thanks.
Okay.
Thank you very much.